Hello friends, today we are going to discuss about advanced techniques in chromatography and their applications. First, we have a look on its objectives, principle and technique involved in ion exchange chromatography, affinity chromatography and HPLC and their applications. Ion exchange chromatography (IEC) is part of ionic chromatography, which is an important analytical technique for the separation and determination of ionic compounds together with ion partition, interaction and ion exclusion chromatography. Ion chromatography separation is based on ionic or electrostatic interaction between ionic and polar analytes, ion present in the eluent and ionic functional groups fixed to the chromatographic support. Two distinct mechanisms as follows: ion exchange due to competitive ionic binding, attraction, and ion exclusion due to repulsion between similarly charged analyte ions and the ions fixed on the chromatographic support. Play a role in the separation in ion chromatography. Ion exchange has been the predominant form of ion chromatography to date. This chromatography is one of the most important adsorption techniques used in the separation of peptides, proteins, nucleic acids and related biopolymers, which are charged molecules of different molecular sizes and molecular nature. The earliest report of ion exchange chromatography date back to 1850. Thomson studied the adsorption of ammonium ions to soil. Spedding and Powell published a series of papers describing practical methods for preparative separation of the rare earths by displacement ion exchange chromatography in 1947. Beginning in the 1950s, Ross and Nelson reported numerous analytical methods which are used for metal ions based on separation on their chloride, fluoride, nitrate or sulfate complexes by anion chromatography. In order to separate proteins, an ion exchange chromatographic method was reported by Peterson and Sober in 1956. In modern form, ion exchange chromatography was introduced by Small, Stephen and Bowman in 1975. When this technique was initially developed, it was primarily used for water treatment. Since 1935, ion exchange chromatography rapidly manifested into one of the most heavily leveraged techniques, with its principles often being applied to majority of fields of chemistry, including distillation, adsorption and filtration. Principal mechanism: ion exchange chromatography, which is designed specifically for the separation of differently charged or ionizable compounds, comprises from mobile and stationary phases similar to other form of column-based liquid chromatography techniques. Mobile phase consists an aqueous buffer system into which the mixture to be resolved. The stationary phase usually made from inert organic matrix, chemically derivative with ionizable functional groups, fixed ions, which carry displaceable oppositively charged ions. Ions which exist in a state of equilibrium between the mobile phase and stationary phases, giving rise to two possible formats. Anion and cation exchange are referred to as counterions. Exchangeable matrix counterions may include protons H positive, hydroxide groups OH negative, single charged monoatomic ions Na positive, K positive, Cl negative, double charged monoatomic ions Ce2 positive, Mg2 positive and polyatomic inorganic ions SO4 2 negative, PO4 3 negative, as well as organic bases Nr2H positive and acid COO negative. Cations are separated on cation exchange resin column and anions on anion exchange resin column. Separation based on the binding of analytes to positively or negatively charged groups which are fixed on a stationary phase and which are in equilibrium with free counterions in the mobile phase according to differences in their net surface charge. Ion exchange chromatography involves separation of ionic and polar analytes using chromatographic supports derivatized with ionic functional groups that have charges opposite that of the analyte ions. The analyte ions are similarly charged ions of the eluent compete to bind to the oppositively charged ionic functional group on the surface of the stationary phase. In modern ion exchange chromatography, the usage of high-efficiency ion exchange materials combined with flow-through deduction have overcome of these challenges. Separations are performed on the column, which is filled with ion exchanger as particles in uniform size. The particles of ion exchange material are generally very much smaller than those used for classical open column ion exchange chromatography. However, ion exchange resins used in modern chromatography have lower capacity than older resins. The eluent must be pumped through the column due to small particle size of stationary phase. The sample mixture is applied into eluent by the injection port. Finally, the separated ions are deducted with a flow-through deduction instrument. The exchange of ions between the ion exchange resin and the solution is governed by two principles. These are: the process is reversible, only rare exceptions are known. The exchange reactions take place on the basis of equivalency in accordance with the principle of electroneutrality. The number of millimoles of an ion absorbed by an ion exchange should correspond to the number of millimoles of an equally charged ion that has been released from the ion exchange. Now, components of ion exchange chromatography. General components of an ion exchange chromatography are presented as a high pressure pump with pressure and flow indicator to deliver the eluent, an injector for introducing the sample into the eluent stream and onto the column, a column to separate the sample mixture into the individual components, an oven optional, a detector to measure the analyte peaks as eluent from the column, a data system for collecting and organizing the chromatograms and data. In ion exchange chromatography, adsorption and desorption processes are determined by the properties of the three interacting entities, which includes the stationary phase, the constituents of the mobile phase, and the solute. Now let us discuss stationary phase. Selection of a suitable ion exchange matrix probably is the most important in ion exchange protocol and is based on various factors such as ion exchanger charge, strength, linear flow rate, sample volume, and sample properties. In ion exchange chromatography, numerous stationary phases are available from different manufacturers which vary significantly in a number of chemical and physical properties. Stationary phase is comprised of two structural elements: the charged groups which are involved in the exchange process and the matrix on which the charged groups are fixed. The charged groups determine the specificity and strength of protein binding by their polarity and density, while the matrix determines the physical and chemical stability and flow characteristics of stationary phase and may be responsible for unspecific binding effects. The next is matrix materials, which includes cellulose hydrophilic surface enhances stability by cross-linking inexpensive. Dextrane considerable swelling as a function of ionic milieu improved materials by cross-linking. Agarose swelling is almost independent of ionic strength and pH. High binding capacity obtained by production of highly porous particles. Polyacrylamide swelling behavior similar to dextrane. Acrylide copolymer high pH stability. Polystyrene divinyl benzene hydrophobic surface low binding capacity for proteins. Coated polystyrene divinyl benzene hydrophilic surfaces. Silica unstable at pH greater than 8 rigid particles. Coated silica hydrophilic surface. Weak and strong ion exchangers. A strong ion exchanger will not lose the charge on its matrix once the column is equilibrated, and so a wide range of pH buffers can be used. On the other hand, weak ion exchangers have a range of pH values in which they will maintain their charge. If the pH of the buffer used for a weak ion exchange column goes out of the capacity range of the matrix, the column will lose its charge distribution and the molecule of interest may be lost. Despite the smaller pH range of weak ion exchangers, they are often used over strong ion exchangers due to their having greater specificity. In some experiments, the retention times of weak ion exchangers are just long enough to obtain desired data at a high specificity. Reasons of ion exchange columns may include functional groups such as weak strong acids and weak strong bases. There are also special columns that have reasons with amphoteric functional groups that can exchange both cations and anions. Examples of functional groups of strong ion exchange reasons are quaternary ammonium Q, which is an anion exchanger, and sulfonic acid S, which is a cation exchanger. These type of exchangers can maintain their charge density over a pH range of 0 to 14. Examples of functional groups of weak ion exchange reasons include diethyl ammonoethyl DEAE, which is an anion exchanger, and carboxymethyl CM, which is a cation exchanger. Now, mobile phase. In ion exchange chromatography, generally eluents which consist of an aqueous solution of a suitable salt or mixtures of salt with a small percentage of an organic solvent are used, in which most of the ionic compounds are dissolved better than in others. Therefore, the application of various samples is much easier. Sodium chloride is probably the most widely used and mild eluent for protein separation due to it has no important effect on protein structure. However, NaCl is not always the best eluent for protein separation. Retention times peak widths of eluted proteins, so chromatographic resolution are affected by the nature of anions and cations used. These effects can be observed more clearly with anion exchangers as compared to cation exchangers. The salt mixture can itself be a buffer, or a separate buffer can be added to the eluent if required. The competing ions, which has the function of eluting sample components through the column within reasonable time, is the essential component of eluting sample. Commonly used eluent additives which have been successfully used in ion exchange chromatography are as follows: EDTA, ethylene diamine tetraacetic acid, polyols, glycerol, glucose and saccharose, detergents, urea and gonadium chloride, lipids, organic solvent, Zwitter ions, sulfahydryl reagents, ligands, protease inhibitor. Buffers. In ion exchange chromatography, pH value is an important parameter for separation and can be controlled and adjusted carefully by means of buffer substances. In order to prevent variation in matrix and protein net charge, maintenance of a constant mobile phase pH during separation is essential to avoid pH changing, which can occur when both protein and exchanger ions are released into the mobile phase. By means of buffer substances, pH value can be controlled and adjusted. Concentration of H positive and the buffering component influence the protein binding to the stationary phase, chromatographic resolution and structure, as well as functional integrity of the protein to be separated. Generally, applications of ion exchange chromatography are performed under slightly acidic or alkali conditions, pH range 6.0 to 8.5, but there are also more acidic and more alkali buffers. Additionally, the buffering component should not act as an eluting ion by binding to the ion exchanger. Ionic buffer components such as phosphate or MOPS in cation exchange chromatography and cationic buffers such as ethanolamine, tris and tricene in anion exchange chromatography are recommended. Typical technique: a sample is introduced either manually or with an autosampler into a sample loop of known volume. A buffered aqueous solution, known as the mobile phase, carries the sample from the loop onto a column that contains some form of stationary phase material. This is typically a resin or gel matrix consisting of agarose or cellulose beads with covalently bonded charged functional groups. Equilibration of the stationary phase is needed in order to obtain the desired charge of the column. If the column is not properly equilibrated, the desired molecule may not bind strongly to the column. The target analytes, that is anions or cations, are retained on the stationary phase but can be eluted by increasing the concentration of a similarly charged species that displaces the analyte ions from the stationary phase. For example, in cation exchange chromatography, the positively charged analyte can be displaced by adding positively charged sodium ions. The analytes of interest must then be detected by some means, typically by conductivity or UV visible light absorbance. Control an IC system usually requires a chromatography data system, that is CDS. Ion exchange chromatography, which is also known as adsorption chromatography, is a useful and popular method due to its high capacity, high resolving power, mild separation conditions, versatility and widespread applicability, tendency to concentrate the sample, relatively low cost. Now, the applications of ion exchange chromatography. The important applications of ion exchange chromatography are as follows. It is extremely used in the analysis of amino acid. The amino acid autoanalyzer is based on ion exchange principle to determine the base composition of nucleic acids. Chergov used this technique for establishing the equivalence of adenine and thymine, guanine and cytosine. This is the most effective method for water purification. Complete deionization of water or a non-electrolyte solution is performed by exchanging solute cations for hydrogen ions and solute anions for hydroxyl ions. This is usually achieved by the method used for softening of drinking water. Proteins are also successfully separated by this technique. It is also used for the separation of many vitamins, other biological amines and organic acids and bases. Now let us discuss chromatography with special reference to affinity chromatography and HPLC. Affinity chromatography separates proteins on the basis of a reversible interaction between a protein or group of proteins and a specific ligand coupled to a chromatography matrix. The technique is ideal for a capture or intermediate step in a purification protocol and can be used whenever a suitable ligand is available for the proteins of interest. With high selectivity, hence high resolution and high capacity for the proteins of interest, purification levels in order of several thousand-fold with high recovery of active material are achievable. Target proteins is collected in a purified concentrated form. Biological interactions between ligand and target molecule can be a result of electrostatic or hydrophobic interactions, van der Waals forces and or hydrogen bonding. To elute the target molecule from the affinity medium, the interaction can be reversed, either specifically using a competitive ligand or non-specifically by changing the pH, ionic strength or polarity. In a single step, affinity purification can offer immense time saving over less selective multi-step procedures. Affinity purification involves three main steps. Number 1. Incubation of a crude sample with the affinity support to allow the target molecule in the sample to bind to the immobilized ligand. Number 2. Washing away non-bond sample components from the support. Number 3. Elution, dissociation and recovery of the target molecule from the immobilized ligand by altering the buffer conditions so that the binding interaction no longer occurs. Now, the principle in brief. The stationary phase is typically a gel matrix, often of agarose, a linear sugar molecule derived from algae. Usually, the starting point is an undefined heterogeneous group of molecules in solution such as a cell lysate, growth medium or blood serum. The molecule of interest will have a well-known and defined property and can be exploited during the affinity purification process. The process itself can be thought of as an entrapment, with the target molecule becoming trapped on a solid or stationary phase or medium. The other molecules in the mobile phase will not become trapped as they don't possess this property. The stationary phase can then be removed from the mixture, washed, and the target molecule released from the entrapment in a process known as elution. Possibly, the most common use of affinity chromatography is for the purification of recombinant proteins. Components of affinity medium. When affinity chromatography is used for the purification and separation of large biomolecules from complex mixtures, the support, that is the matrix, spacer arms and ligands must be considered. Applications of affinity chromatography. Affinity chromatography can be used in a number of applications, which includes nucleic acid purification, protein purification from cell-free extracts, and purification from blood. Now, discussing a brief about high-performance liquid chromatography, which is also known as HPLC. High-performance liquid chromatography, HPLC, formerly referred to as high-pressure liquid chromatography, is a technique in which analytical chemistry uses to separate, identify, and quantify each component in a mixture. It relies on pumps to pass a pressurized liquid solvent containing the sample mixture through a column filled with a solid absorbent material. Each component in the sample interacts slightly differently with the absorbent material, causing different flow rates for the different components and leading to the separation of the components as they flow out the column. HPLC has been used for manufacturing, for example, during the production process of pharmaceutical and biological products. Legal, for example, detecting performance enhancement drugs in urine. Research, for example, separating the components of a complex biological sample or of similar synthetic chemicals from each other. And medical, for example, detecting vitamin D levels in blood serum purposes. Chromatography can be described as a mass transfer process involving adsorption. HPLC relies on pumps to pass a pressurized liquid and a sample mixture through a column filled with adsorbent, leading to the separation of the sample components. The active components of the column, the adsorbent, is typically a granular material made of solid particles, for example, silica, polymers, etc. 2 to 50 micrometers in size. The components of the sample mixture are separated from each other due to their different degrees of interaction with absorbent particles. History and development. Period to HPLC scientists use standard liquid chromatographic techniques. Liquid chromatographic systems were largely inefficient due to the flow rates of solvents being dependent on gravity. Separations took many hours and sometimes days to complete. Gas chromatography (GC) at the time was more powerful than liquid chromatography (LC). However, it was believed that gas phase separation and analysis of very polar high molecular weight biopolymers was impossible. GC was ineffective for many biochemists because of the thermal instability of the solutes. As a result, alternative methods were hypothesized which would soon result in the development of HPLC. Following on the seminal work of Martin and Singe in 1941, it was predicted by Carl Giddings, George Huber and others in 1960s that LC could be operated in the high efficiency mode by reducing the packing particle diameter substantially below the typical LC and GC level of 150 IM and using pressure to increase the mobile phase velocity. These predictions underwent extensive experimentation and refinement throughout the 60s and the 70s. Early developmental research began to improve LC particles and the invention of ZPEX, a superficially porous particle, was promising for HPLC technology. The 1970s brought about many developments in hardware and instrumentation. Researchers began using pumps and injectors to make a rudimentary design of an HPLC system. Now let us discuss types of HPLC system in brief. These are following variants of HPLC depending upon the phase system, stationary in the process. Number 1 is normal phase HPLC. This method separates analytes on the basis of polarity. Normal phase HPLC uses polar stationary phase and non-polar mobile phase. Therefore, the stationary phase is usually silica and typical mobile phases are hexane, methylene chloride, chloroform, diethyl ether and mixtures of these. Polar samples are thus retained on the polar surface of the column packing longer than less polar materials. Number 2 is reverse phase HPLC. The stationary phase is non-polar, that is hydrophobic in nature, while the mobile phase is a polar liquid such as mixtures of water and methanol or acetonitrile. It works on the principle of hydrophobic interactions, hence the more non-polar the material is, the longer it will be retained. Size exclusion HPLC is another type. The column is filled with material having precisely controlled pore size and the particles are separated according to their molecular size. Larger molecules are rapidly washed through the column. Smaller molecules penetrate inside the pores of the packing particles and elute later. The next one is ion exchange HPLC. The stationary phase has an ionically charged surface of opposite charge to the sample ions. This technique is used almost exclusively with ionic or ionizable samples. The stronger the charge on the sample, the stronger it will be attracted to the ionic surface and thus the longer it will take to elute. The mobile phase is an aqueous buffer where both pH and ionic strength are used to control elution time. Instrumentation. HPLC instrumentation includes a pump, injector column, detector and integrator or acquisition and display system. The heart of the system is the column where separation occurs. Lastly, applications of HPLC. The information that can be obtained by HPLC includes resolution, identification and quantification of a compound. It also aids in chemical separation and purification. The other application of HPLC includes pharmaceutical application, which in turn includes to control drug stability, tabular dissolution study of pharmaceutical dosages, pharmaceutical quality control. Environmental application includes detection of phenolic compounds in drinking water, biomonitoring of pollutants. Application in forensics include quantification of drugs in biological samples, identification of steroids in blood, urine, etc. Forensic analysis of textile dyes, determination of cocaine and other drugs of abuse in blood, urine, etc. Food and flavor includes measurement of quality of soft drinks and water, sugar analysis in fruit juices, analysis of polycyclic compounds in vegetables, preservative analysis. Application in clinical tests includes urine analysis, antibiotic analysis in blood, analysis of biliverdin in hepatic disorders, detection of endogenous neuropeptides in extracellular fluid or brain, etc. So friends, this is all about advanced techniques in chromatography and their applications. We will be meeting again. Till then, goodbye and stay blessed.